So this is next week's paper. Opening and closing of a bacterial RNA polymerase clamp. Scary. Science, is this the first science paper we've read, or have we done a science paper? They're nice and short. This will be a good contrast to the paper we just read, and then to the paper the next week, which is like 21 pages long or something. But it's really well written. Yeah. So enjoy it this week, because it's only 4 pages. Okay. Okay. So here's the few things that I think you should probably know before you read the paper. Fret, an experimental approach called FRET. Don't fret, it's not that hard to understand. Which requires you to know something about 4-azetophenylalanine and how they incorporated probes. We'll go over those. In vitro reconstitution of core RNA polymerase. Talk about what that means and the difference between core polymerase and hollow enzyme. And then the steps of transcription initiation. So the point is, on the syllabus for next week, we're doing prokaryotic transcription. That's the topic that I'm supposed to cover next week. This is a paper about bacterial transcription. I think this is a pretty good paper, but as I mentioned at the start of class, it's very different. It's a structural paper, so it's biology, but it's also chemistry. Structural chemistry. So that's why we're talking about RNA polymerase, right? The enzyme that does transcription. And the point of this paper is, well, we'll get to the point later. Let me talk about this first. FRET, fluorescence resonance energy transfer. What? People use FRET to do everything. FRET's really cool. In this case, what we're trying to do is, there's a part of RNA polymerase that looks like a big crab claw. It's called a pincer. And guess what goes in there? DNA. For transcription. And the question basically is, at different steps during transcription, is this open, closed? How open is it? How closed is it? What's the conformation of this part of the protein? So it's a structural paper, trying to figure out what this protein's shape looks like at different steps of transcription. And you can quantify that if you know, for example, how far apart are the tips of the pincer at different steps in transcription? So basically, this whole paper is about measuring the distance between those two parts of this one protein, RNA polymerase. Are they close together or are they far apart? That's where FRET comes in. So if you attach two fluorescent molecules, one on one side of the protein, one on the other side, these are fluorescent molecules. And what this shows is that when light, let's see, hey look, light. When light comes in, it excites this fluorescent molecule, right? This is how fluorescence works. Light hits, what does the fluorescent molecule do? It absorbs it. Yeah, it emits light. It absorbs one wavelength, and then it emits a different wavelength of light. The way FRET works is that wavelength of light that's emitted, that's the energy transfer part. So fluorescence, this molecule is fluorescing. It's transferring energy in the form of a specific wavelength of light, the emission wavelength, that happens to be the excitation wavelength for the other fluorescent molecule. And then that molecule is excited by the fluorescence given off by the first molecule, and it gives off a second emission wavelength. So you know that if these two, the closer together in physical space these two molecules are, the more likely it is that the light, the fluorescence that's emitted from the first probe excites the one that's next to it, because they're closer together. It's more likely that that light's going to hit the next fluorescent molecule, and it gives off the other wavelength of light, right? A longer wavelength. And then that's what the scientist detects. How much light is being given off by the second fluorescent molecule. that tells you something about how close together those two molecules are in space. So another way to look at that is this graph. It says exactly the same thing. The donor molecule, the donor probe, has a certain excitation wavelength. So you shine light of that wavelength. It emits that spectrum of light. And so part of its emission spectrum overlaps with the excitation spectrum of the next molecule. And then it gives off a specific wavelength of light that you detect. You shine light in, you expect to get this wavelength out if those two molecules are very close together. The farther apart they are, the less light you get out. Sorry, I can only find a German version of this figure, so please ignore the German.
Intensität.
Yes, the Intensität in the donor and the receptor, ja. Jawohl. And lambda x and lambda m, which apparently are the same in German and English. Anyway, so that is FRET. So that's how they're using it. They want to make RNA polymerase that has two specific fluorescent molecules at specific points on the protein. And then they assemble the whole RNA polymerase enzyme, and then they do these measurements. Right? So, for azitophenylalanine, they mention in the paper, it's critical, but it's a small part of the paper. It's an unnatural amino acid. It's a synthetic amino acid. Here's the amino acid part. And normally there'd be amide bonds formed here to one amino acid and there to the next. So this is the direction the protein would be going. This is the side chain. So it's a phenylalanine with 4 azido on it. Unnatural amino acid.
So just a benzene ring and NH3.
Yeah, N3.
N3?
Yeah.
Sorry.
That's okay. Yeah. So it's a type of phenylalanine. And I'm not going to go through how they incorporate this amino acid into the protein, which is a whole other thing that would take a while to talk about. But just know that they have a mechanism where they can put this amino acid in a specific spot in the protein. So that gets us part of the way there. They have phenylalanine in two parts of the protein on both sides of the claw. Now they need a way to stick, and the reason they're doing this is because that N3 group is highly reactive. So they can use that to covalently bond those two fluorescent molecules onto the two different parts of the protein. And that's this process that you'll also read, the Staudinger ligation. Oh, I wish I'd paid more attention to organic chemistry. It doesn't really matter. The way this works is you have any molecule, oops, sorry, I should use this. You have any molecule that has that N3 group on it, just like that 4-azitophenylalanine. And that, in certain reaction conditions, will react with an organic molecule to ligate that organic molecule right there on that N. So it's just a reaction that you can use to covalently bind. So presumably this figure is just some random figure I got from Google Images. This has nothing to do with this paper. But in the paper, this molecule would be the fluorescent probe. And they're going to attach it using this reaction to that N3 group that's in the unnatural amino acid that's incorporated into the protein. So it's 4-azitophenylalanine covalently bound by this reaction to fluorescent molecule A on one side of the claw and fluorescent molecule B on the other. So assuming you can do all that, and it works, that's not the end of the story. You've got RNA polymerase, which has a huge number of protein subunits to make up an actual complex that can transcribe DNA. It's not just one protein. So terms you need to know: core polymerase, 5 subunits. Alpha, alpha prime, beta, beta prime, and omega. So you have to put 5 proteins together to make up something that looks like RNA polymerase. Right? They all bind together and make the core polymerase. What's missing in core polymerase, as it says here, is there's no sigma factor, another protein, sigma factor. And that core polymerase doesn't do transcription. You have to have sigma factor and you have to have DNA to do transcription. That's what the hollow enzyme is. So when they say core, they're talking about those 5 subunits without sigma factor, without DNA. So what they're going to do is they're going to see what is the structure of this when it's just core enzyme, and then how does it change when you add sigma factor and DNA? Because that's when this protein, this enzyme, would actually start synthesizing. Then there's the in vitro constitution, which basically means they make all of these proteins in vitro, in tubes, separately, and they just mix them all together and assume that they all combine together in a tube like they would in the cell to make RNA polymerase.
Just as a clarification.
Yeah.
So in cell biology, in vitro is outside the cell.
Yes.
And in vivo is in the cell, but in other fields,
Right.
in vivo is in animal model versus in vitro being in cells.
Yeah, that's horrible, but I agree.
It's confusing like that.
This is real. This is in vitro.
Okay.
So they're doing exactly what I said. They make.
Wait, in vitro? As in.
Sorry,
it's
not twice.
It's not a living organism. Okay.
In a tube.
In a tube, yeah.
Yeah. So you take a plasmid. Yeah, like you take a plasmid that contains the gene for the sigma subunit, and you in vitro transcribe, which means you take extract of these cells and this thing from those cells and a mixture of amino acids, and in that tube you get a bunch of that one protein. And then you combine alpha, alpha prime, beta, beta prime, omega, sigma, and some DNA together, and you just mix them all together in one tube, and you hope that they come together the way they're supposed to. We'll see. Okay. Steps of transcription initiation. Yeah, these are important because this is the bulk of the paper, is measuring this at all of these different stages. So you start, step 1 is just core. Right, as we just described, those 5 subunits, nothing else. What's the conformation of the protein? Add sigma 70 and you get the hollow enzyme. Add DNA, and these are the terms that you need to know for next week. RPC, you don't need to memorize them, of course, but these are things that the authors refer to a lot, those acronyms. PC, step 3, promoter closed. So if you look at the picture in step 3, you see the enzyme's bound on DNA, but the DNA has not unwound yet. It's closed. It's all double-stranded still. Then the next step is that the polymerase actually melts a little bit of the DNA, makes it single-stranded. You get that bubble right there. So that's RPO, RNA polymerase promoter open, PO. Then the next step after that is it starts to synthesize small amounts of RNA. That's the ITC, initial transcribing complex. And then at some point, the sigma factor releases and the polymerase just starts shooting down the DNA, synthesizing RNA. And that's RDE, the last step. And don't forget, I post all this online, so definitely write it down. It's good for the memory, but this is available to you as well. Okay, so these are the different steps at which the authors are going to be asking what's the distance between the 2 tips of the pincer. Questions?
So the pincer on the diagram?
Yeah, it's that,
And then
it looks like that, bound around the DNA. It's those. And really, the only reason they want to know is because no one's ever looked at this before and they have the opportunity to do it. There's a big question, what's the structure of polymerase as it synthesizes? So they're going to do this cool thing that got them a, what was this, Science or Nature? Got them an awesome paper, Science. Yeah. Okay. I decided to throw this in because every field has some sort of crazy terminology. They talk about scrunching. RNA polymerase initially transcribes by scrunching. And that basically means that the first thing that RNA polymerase does here is it actually, it doesn't move on the DNA. So you'll notice that the back of RNA polymerase stays at minus 35 to minus 10. So the polymerase itself is not moving on the DNA. But it pulls some DNA inside itself. So the DNA sort of scrunches up inside the polymerase, shown there. So that's presumably the first step of synthesis, is the polymerase doesn't move anywhere, it pulls the DNA into it and starts synthesizing. Okay. So that's what they mean by scrunching. It's, like I said, it's not important, I just thought it was kind of funny. Every field has its own weird terminology. This is what the figures are going to look like. Histogram, so frequency on the y-axis, and what's this? Fret efficiency. What does that mean to us? Yeah, amount of fluorescence this direction. So the closer the two things get, the higher the efficiency of transfer. That is, the more energy is being transferred directly to the acceptor, not shot out into space.
And what's on the y-axis?
Frequency, so number of observations. So what they're doing, as I understand it, although I'm not an expert, of course, in vitro reconstitution of enzymes, is they put all these things together, so they've got all these complexes floating around in solution, and they're shining a laser into the solution. And every time one of these complexes goes through, the laser is exciting that first molecule, it's that wavelength of light. So when a molecule floats past the laser, it gets excited, then they're looking for emission of that light that comes out of the acceptor. So every time a protein goes past the laser and gets excited, they detect one measurement. Did it excite, did we get light out or did we not? So they just keep shining that light in the solution, and hundreds and thousands of complexes just happen to drift by the laser, and so every time that happens they detect, was there light and how much? So this is a histogram of all of the observations. Let's say this is 10,000 observations. Of all of the observations, how many had an efficiency of 0.5? Some fraction of the total observations gave that much light out when they were excited. So this is a distribution of how close, because presumably in solution, even if this is at one step in transcription, there's still a bit of flexing in the protein. So you're not going to get exactly the same, at one step in transcription, you probably don't get exactly that amount of light out of every complex that you look at. So you get a distribution where there's a mean. So the peak of the distribution, right, presumably, and please feel free to argue about this after you've read the paper next week, presumably the peak of the distribution is the true, most frequent distance between the tips of the pincers. There are some complexes down here where there was, at this step in transcription, no efficiency. Looks like they're way out here like this. And there were some molecules way up here where it looks like they were right next to each other. Probably experimental error. We'll talk about that. They talk about that in the paper. We'll talk about that next week. So presumably the tallest part of the peak here represents exactly how far apart most of the complexes are.
Did we see something like that a couple weeks ago too?
We have seen some things that look like this before. Yeah, more histograms. Yeah. So these are the expected values of efficiency for certain of those steps in transcription, RPO, RPC, RITC, whatever.